Hey there, welcome back. For today's video, we're going to demonstrate the proper way of doing the aseptic technique and the strict plate method. But before we advance to the procedures, let us define them first. Aseptic technique is the use of different routines to prevent contamination of a culture, sterile media stock, and other solutions from unwanted microorganisms or pathogens. On the other hand, the strict plate method is a procedure that uses simple mechanical separation to isolate pure cultures of bacteria or colonies from mixed populations. With this, a mixture of cells is spread over a surface of a semi-solid, agar-based nutrient medium in a petri dish, such that lesser and lesser of the cells are piled up at points far away from each other on the medium surface, and after incubation, develop into colonies. Now, let us show you the proper way of doing these. To start, the materials required for this are petri dish with appropriate bacterial media, inoculating loop, Bunsen burner or alcohol lamp, marker, masking tape, and your chosen microorganism. We will execute the aseptic technique first by preparing a safe and sterile workplace. 1. Familiarize yourself with the laboratory rules and safety precautions to be observed when handling microorganisms. In line with the rules, do not forget to wear the proper attire for working in a laboratory. 2. All instruments, solutions, and media prior should be sterilized before using them for the plating. 3. Remove all the materials blocking your workspace. 4. Using 70% volume per volume ethanol, clean your work area. 5. Set up the alcohol lamp and work cautiously near the flame it creates, which is a sterile field. 6. All things needed for the process shall be properly arranged near the sterile field. The Bunsen burner should be placed to your right while the petri dishes to your left. Cell cultures, tubes, flasks, and bottles shall be positioned in the center of the bench. 7. Be certain that your hands are properly washed with antiseptic soap and warm water. That is all for the aseptic techniques. Let us now proceed to the strict plate procedure using the quadrant method. 1. Label with your name, the date, the type of growth medium, and the type of organism to be plated the edge of the bottom of the petri dish. Note that plates should be completely dry and pre-warmed to room temperature. 2. Flame the inoculating loop using the alcohol lamp until it becomes red hot. 3. Obtain the inoculum for the plate. If the inoculum is a cell suspension, get a loop full with a metal loop. The rim of the tube should also be flamed before and after removing the inoculum, just like the loop. If the inoculum is a colony, use a cooled loop to touch the colony and be able to get a few cells from it. 4. For the first quadrant, by holding the loop like a pencil at a 10 to 20 degree angle to the medium, spread the sample at about 1/4 of the surface of the medium using a rapid, smooth, back and forth motion. Do not forget to flame the edge of the petri dish before and after streak. 5. Flame the metal loop again and turn the petri dish 90 degrees to streak the second quadrant. Use the loop to touch somewhere near the last streak of the first quadrant. Then, with the same pattern, move to the second quadrant. Close the lid when the second quadrant is filled. 6. Repeat step 5 for the third and fourth quadrants. The metal loop should always be reflamed in between each quadrant, and in streaking the fourth quadrant, avoid touching the first quadrant again. 7. Incubate the plate upside down so that the condensation of the lid will not fall into the colonies. Also, don't forget to use 2 strips of tape to seal the Petri dish, and that's it. Thank you for watching. We hope that you have learned about the aseptic technique and the streak plate method. Don't forget to give us a thumbs up. Until next time!